The flu is not a syncytial virus. The flu has to leave a cell to infect another cell. And when it leaves that cell, it's vulnerable to your immune system. As long as it's inside a cell, and this is the problem with HIV, is that HIV gets into cells that does not undergo apoptosis and do not form syncytia. So when syncytia gets formed, this is a really important topic to understand in the midst of this conversation. When syncytia is formed, syncytia is not something the virus necessarily does, right? It's something that our cells do, and COVID or other pathogens hijack that mechanism. So this is what makes pathogens on a risk scale depends on their ability to hijack our various mechanisms. And if they can hijack the mechanism that our cells use to make syncytia, then they are the highest order of risk. That is why COVID is the highest risk, will never be flu or colds, because it has the ability to hijack our cells' ability to form syncytia. That made sense finally, right? And so a lot of people think of this as something the virus does, and it's actually something our cells do. And actually, syncytin, I wrote a thing about this today. This is very, very interesting because it's all related to why we don't hatch in eggs. So hundreds of thousands of years ago, we got infected with a virus that gave our cells the ability to fuse cells. So we did gain it from a virus like a really, really long time ago. And it's the primary process by which the fetus through the umbilical cord and the sac fuses to the uterus so that you can feed the baby.
Wow.
That fusion happens through a thing called syncytin because those cells permanently fuse. And I have a theory, my long-standing theory has been that the reason the blood clots look like cell masses that kind of remind you of like birthing bits. I don't know what the right word is for that, but that's, all that sort of stuff, because it's all undergoing that syncytia process. And so that material is how the syncytia forms in our normal cells. So when it bursts, all that stuff, including the syncytia, goes loose into our system, and it causes cells to fuse into these big, long, glumpy structures that don't break down under normal anticoagulants because they are not coagulated blood. They are cell structures of waste that have fused together post syncytia bursting.